Okay, so in this exercise here, we are going to do a pathway analysis based on the gene patterns or the mutational patterns that we have identified from the previous video. So what we did previously is that basically if you open up the predictor or the classifier data here. So for example, KNN mutation, what we have here is that the accuracy is 0.611, and then these are the classifier altogether, 10 of them. And then if you look at SVM, it's quite well, it's much better, it's 0.9722 the accuracy, but the genes list is actually, because classifier is actually quite long, and then you also have another one which is using a random tree. So what we are going to do now is basically we summarize all these here. So these are the classifier based on KNN modeling, and then these are the one based on SVM, and these are the one based on random tree. So to what now what we are going to do is we are going to overlap it and to look at the common uh, classifier that is predicted by tree model. So to do that, we can go into a Venn graph. You can go into this address here, which is a very nice online software that will help you to draw a Venn graph. So what we are going to do here is we are going to import the KNN list here, and then we can label this as KNN, and then we can then continue on with SVM. So and also continue this on with RT, the other random tree. Okay, so what you will get, then you click submit. Okay, and here's what you will get. Basically, what you will find is that the random tree and also the KNN, they all the classifier that's identified are all consistent between the KNN and also the random tree model. Whereas SVM, you also have the same 10 genes which is in the SVM classifier, but the other 189 was only unique to the SVM. So to choose those classifiers that are most stable, we will basically choose uh, based on this region here, whereby these are the classifier that is predicted by tree model. So in this case here, so this is the genes that we might be interested in to look at, okay, whether it have any biological function that might explain why the gemcitabine is not responding I mean, why the tumor with this mutation are not responding to gemcitabine. Okay, what I'm going to do next is basically to look at what are these 10 classifiers that we identify, what is a biological function, and what are the pathway that it might be um, affected. So there are a couple of software that you can run the pathway analysis. Now I'm going to show you a few of those. Now the good things about it is that almost all this can be done online, so you can actually do it by connecting to the database and also the online software. Now, so what I'm going to do now is I'm going to just copy the 10 stable classifiers that we have identified from the mod for the model. And the first thing I'm going to try, I'm going to show you to use a string, which is well, you can go to this website here. So it is actually quite interesting software to look at. So now you can go to multiple proteins, and then you just copy and paste the gene list here, and then select the organism, which is Homo sapien, and then it will just search. So okay, it will automatically map it to the database. So and then you just click continue. Now, so in this case here, okay, if there is two nodes that are actually connected by a line, that means that, well, somehow the function or actually they are related to each other in terms of function, and as well as other, what you call it, gene co-occurrence and so forth. Now in this case here, it's quite interesting to see is that while most of this mutation basically is actually isolated, meaning that they are not related to a particular a subset of the pathway. So except for GNAS and also ATCY8. So this is quite interesting. So anyway, so if you click on this button here, it will give you a snapshot or some information about the genes that is and the function that it is related. And also if you look at ATCY here, then it will show you as well. But the bottom line is that, well, it seems that, well, the classifier that we identify and the mutations that are associated with gemcitabine are actually quite independent to each other. So next, we are going to try another software which is called the Reactome pathway. It also is a huge database that actually curated to look at the pathway that have been reported in the public domain, meaning the literature or journal publications. So here you can go to this UTL here, reactome.org. So what you need to do is you go to analyze data here. Okay. So again, well, you can just paste the gene list here, the 10 classifier that you identify, and then you click continue. Now, here you can actually have the option to select, well, whether you want to include the interactors, okay, to look at your pathway that might be enriched by certain the mutation that you just in, just input into the analysis. So now the interactors is quite interesting because if the genes is mutated, so it is expected that it might also affect the function of the protein that is interacting with them. So, well, you can play around and you can actually include these in, but for our current analysis, okay, so we will just stick with the default, which is just to look at the genes that we are interested in. So basically, with them, you just click analysis here. Then straight away, well, they will tell you basically, okay, in your gene set, what are the common pathway that is particularly enriched. For example, in this case here, the PKA activation in glucagon signaling basically have a very low p-value, meaning that, well, the genes that you are in the list of the genes that you are inputting, a lot of it are actually involved in the glucagon signaling. So, and not surprisingly, also is that, well, there's another two more subsets which is also associated with it. What it suggests here is basically, okay, the mutation of the genes that are involved in glucagon signaling might render the cells to become resistant to gemcitabine, which is somewhat quite interesting. So you might want to then look into the literature and see how this could be explained or have whether it have been reported or not. So one cool thing is that, well, you can actually click on the button here, okay, and then it will automatically zoom in for you. And if you want to look into this node, then you can actually zoom in further and to show you basically what are the genes that is being affected inside the pathway. So for example, okay, if you look at the glucagon signaling here, so it will show you exactly in where your gene mutation are actually affecting the function of that particular pathway. So we'll wait for a while for the diagram to load. So you can also scroll down here. There are also other pathway which might be affected by the gene mutations that you have identified as well. Okay, so it will take a while to load it. So okay, so anyway, so we would, oh, okay, here you go. So if you look at it, see, okay, so this blue line is basically where your gene mutations are actually affecting it. For example, this color, green color uh, things here are basically showing that the the genes that you have input while you had input into the gene list just now, you can see that, okay, most likely is that within the glucagon pathway. So these are the node or these are the biochemical mechanism that your mutation might be affecting it, and how this will affect the gemcitabine response, then that will serve as a hypothesis that could be further tested in the in vitro or the in vivo model. So there are also a couple of other software that you can play around, so including David Bioinformatics, so you can actually look and learn how to do it, or you can also use the consensus path DB. In this case here, well, this is the consensus path DB is good in the sense is that, well, it can actually analyze simultaneously all the database that is currently known in terms of pathway analysis. So to do that, I'm just quickly demonstrate how you can do it. You can go to gene set analysis, over representation analysis. You just paste the list here, select. So this is the gene symbol, and then you click proceed. So you can actually look at the gene ontology if you want to, but I more prefer to look at the pathway by the pathway database. Here is where you can actually select what database that you want to look at. So the common known ones, for example, KEGG and also Reactome. Again, well, this will then link it to the Reactome pathway that we have used just now. So there are a couple of other pathway database that it will also compare as well. So it's a very powerful and very simple software. So if you set it here again here, what you can see here is that, okay, so based on the PID pathway, so you have uh, well, the mutations are highly associated with this LPA4 mediated signaling events. I do not know what it is. You can read about it. But based on the reactome, you see again, this tree was actually the highest in terms of statistical significance, okay, as predicted by the reactome pathway as well. So with this one here, you can actually, in one shot, you can actually compare it and analyze it in different database. So that's it for now. So I hope you enjoyed this. So and do play around it. So and look into, okay, how and what are these glucagon pathway might be associated with the gemcitabine response. That's all for now. Thank you.